Good day everyone, I'm Shane Batacan and I will going to discuss the introduction, function, and action of enzymes. So what is enzymes? An enzyme is a biological catalyst and is almost always a protein. It speeds up the rate of a specific chemical reaction in the cell. The enzymes is not destroyed during the reaction and is used over and over. A cell contains thousands of different types of enzyme molecules, each specific to a particular chemical reaction. Did you know that enzymes speed up chemical reactions? In some cases, enzymes can make a chemical reaction millions of times faster than it would have been without it. The chemical reactions that keep us alive, our metabolism rely on the work that enzymes carry out. A substrate binds to the active site of an enzyme and is converted into products. Once the product leaves the active site, the enzyme is ready to attach to a new substrate and repeat the process. So let's proceed to the function of enzyme. How do enzymes speed up biochemical reactions so dramatically? Like all catalysts, enzymes work by lowering the activation energy of chemical reactions. Activation energy is the energy needed to start a chemical reaction. Next is the mechanism of enzymatic action. First is the attracts. An enzyme attracts substrate to its active site, catalyzes the chemical reaction by which products are formed, and then allows the product to dissociate or separate from the enzyme surface. Next is the combination. The combination formed by an enzyme and its substrate is called the enzyme-substrate complex. Another one is the binary complex. When two substrates and one enzyme are involved, the complex is called a ternary complex. One substrate and one enzyme are called a binary complex. Another one is substrate are attracted to the active site by electrostatic and hydrophobic forces. And lastly, the physical, which are called non-covalent bonds because they are physical attractions and not chemical bonds.
Good day everyone, I'm Christine Joy de Concepcion from BSED 2D. And I'm going to discuss what do enzymes do and how does it works. First is what do enzymes do. One of the most important rules of enzymes is to aid in digestion. Digestion is the process of turning the food we eat into the energy. First, let's talk about the digestive system. Enzymes create chemical reaction in the body and are crucial for the variety of processes including digestion. Enzymes help the body break down larger complex molecules into smaller molecules, such as the glucose, so the body can use them as the fuel. Your saliva has digestive enzymes in it. Some of your organs, including your pancreas, gallbladder, and liver, also release them. The second one is all about DNA replication. Each cell in the body contains DNA. Each time a cell divides, that DNA needs to be copied. Enzymes help in this process by unwinding the DNA coils and copying the information. DNA replication is a very complicated process that involves several enzymes and other proteins. It occurs in three main stages. The first one is initiation, second is elongation, and the third is termination. The last one is all about liver enzymes. The liver breaks down toxins in the body, but to do this, it uses a range of enzymes. Enzymes are proteins found in your body that speed up certain chemical reaction. Liver enzymes perform this job within the liver. Elevated liver enzymes are the sign that a person has an inflamed or damaged liver, and many conditions may cause liver inflammation or damage. The next one that I will discuss is how enzymes work. The lock and key model was first proposed in 1894. In this model, an enzyme's active site is a specific shape and only substrate will fit into it like a lock and a key. This model has now been updated and is called induced fit model. In this model, the active site changes shape as it interacts with the substrate. Once the substrate is fully locked in and in the exact position, the catalysis can begin. It also has three parts. First is the perfect condition. Enzymes can only work in certain conditions. Most enzymes in the human body work best at around 37 degrees Celsius body temperature. At lower temperature, they will still work but much more slowly. Similarly, enzymes can only function in a certain pH range, acidic or alkaline. Their preference depends on where they are found in the body. For instance, enzymes in the intestine work best at 7.5 pH, whereas enzymes in the stomach work best at pH 2 because stomach is much more acidic. If the temperature is too high or if the environment is too acidic or alkaline, the enzymes changes shape. This alters the shape of the active site so the substrates cannot bind to it. The enzymes has become denatured. Next is the cofactors. Some enzymes cannot function unless they have a specific non-protein molecule attached to them. These are called cofactors. For instance, carbonic anhydrase, an enzymes that help maintain the pH of the body, cannot function unless it is attached to a zinc ion. And the last is inhibition. To ensure that the body system works correctly, sometimes enzyme needs to be slowed down. For instance, if an enzyme is making too much of a product, there needs to be a way to reduce or stop production. Enzymes activity can be inhibited in a number of ways. First is the competitive inhibitors. It is a molecule blocks the active site so that the substrate has to compete with the inhibitor to attach to the enzymes. The second one is the non-competitive inhibitors. It is a molecule bind to an enzyme somewhere other than the active sites and reduce how effectively it works. The third one is the uncompetitive inhibitors. The inhibitor binds to the enzyme and substrate after they have bound to each other. The product leaves the active site less easily and the reaction is slowed down. And the last is the irreversible inhibitors. An irreversible inhibitor binds to an enzyme and permanently inactivate it.
Let's talk about examples of a specific enzyme. Let's start with the amylase. So amylase is an enzyme that helps us digest our food. They breaks down the starch, turning them into sugar. And amylase can be found in a human saliva, and it is also present in some other mammals like koala and kangaroo. Next is lipase. Lipase are a group of enzymes that is made by our pancreas, and it helps our body in digesting fats. Obviously, it is located in our pancreas because that's the place where it is being produced. Maltase is an enzyme that can be found in plants, bacteria, and it is also present in foods like potatoes, pasta, and beer. Maltase breaks the sugar maltose, turning them into glucose. Lactase is an enzyme that helps to digest lactose, which is a sugar that can be found in milks. It breaks lactose, turning it into glucose and galactose. And lactase can be found in our small intestine. Trypsin, an enzyme that helps us to digest proteins by breaking it, turning it into amino acids. Trypsin can be found in our small intestine. Acetylcholinesterase, an enzyme primarily found at postsynaptic neuromuscular junctions or in the middle of the fiber. They breaks down the neurotransmitter acetylcholine in our nerves and muscles. Helicase. are molecular motor protein that manipulates our DNA and RNA. Their main function is to unpack an organism's genetic material. DNA polymerase synthesize the DNA from deoxyribonucleotides, and their primary role is to accurately and efficiently replicate the genome in order to ensure the maintenance of the genetic information and its faithful transmission through generations.
Good day everyone. I am Danica Abuan Macanas from BS Ed 2D. And the part or the topic that I'm going to discuss is all about enzyme deficiencies. So when we say enzyme, it is a special type of protein needed to break down the food molecules so that it will turn into fuel during metabolism. So when we say deficiency, it is the absence of something. So if we're going to correlate or combine the word enzyme and deficiencies, enzyme deficiencies is the absence of the enzymes. Moreover, enzyme deficiencies are inherited defects that may result in a number of life-changing or life-threatening conditions. So meaning to say, enzyme deficiencies can be passed down from the parents to their offspring or to their children. So are you familiar with the word mucopolysaccharidosis or MPS? This is a group of inherited diseases wherein there is defective or missing enzymes that causes complex sugar molecules to accumulate in cells. And just like what I said earlier, mucopolysaccharidosis are a group of inherited diseases, and those inherited diseases are the lysosomal storage disorders. And lysosome function as the primary digestive units within cells. And the enzyme within the lysosome is responsible for breaking down nutrients such as carbohydrates and fats. Additionally, lysosomal storage disorders contains about 50 inherited diseases that occurs when there is a missing enzyme, results in the body's inability to recycle the cellular waste. Moreover, lysosomal storage diseases results to the dysfunction of the organ because the lysosome within the cell are defective because of the enzyme within that is responsible for breaking down nutrients molecule. So here is the example of enzyme deficiency. So this is 9-man break. This is a rare inherited disease that has effect on the ability of our body to metabolize fat such as cholesterol and lipids. So what will happen to the cell? The cell will malfunction and later will die. So what are the human body organs that are affected by this disease? Those are brain, nerves, liver, spleen, and also bone marrow. And also others like lungs. So the patients that are diagnosed with this kind of disease lacks of a critical enzyme which is necessary to metabolize fatty substances in the body called lipids. So that's all for enzyme deficiencies. Once again, I am Danica Aban Macanas from BSL 2D. Thank you.